Hey guys, welcome to this Mometrics video on the basic characteristics of an organism. Before we dive in too deep, let's define an organism. Organism comes from the Greek word organon, which literally means instrument. In short, an organism is anything that is living, senses, experiences growth, reproduces, and works to regulate internal conditions. But like I said, that is in short. Let's go a little deeper and look at the 7 characteristics of a living organism. 1. Nourishment. All living things are dependent on external resources to grow, repair, and to maintain current internal processes. So, an organism must take in nutrients to obtain the energy it needs to maintain all of its functions. 2. Respiration. Now, we need to be sure not to confuse respiration with breathing. Breathing is the more physical process, where we inhale oxygen and exhale carbon dioxide. Respiration is a more chemical process. However, breathing is very important for respiration to take place. Respiration is when oxygen and glucose combine to produce carbon dioxide, water, and ATP. Something that is interesting to note is that autotrophs, or an organism that is photosynthesizing, are dependent on heterotrophs, organisms that are not photosynthesizing, like humans, and vice versa. 3. Motion. Every living thing moves. This may seem like an obvious observation with organisms like humans, animals, insects, but plants move as well. And I'm not talking about them being moved by the wind. They actually move on their own. Leaves turn to face the sun, flowers open and close, and climbing plants literally reach out until they can grab onto something. 4. Expel. With all these nutrients coming in and being used for energy, the waste has to go somewhere, out. In fact, the waste from the chemical reactions that go on within the organism can actually be toxic to the organism, so it must be excreted or expelled from the organism. So all organisms excrete or expel waste. 5. Grow. All living organisms grow. Growth in an organism can be defined as the constant increase of size and the number of cells. 6. Reproduce. Every organism has the capability to produce offspring. Living things multiply. 7. Sensing. Sensing refers to the ability every organism has, and that is to sense and to react to stimuli. Stimuli include things like chemicals, water, gravity, light, and temperature. Hey guys, welcome to this video on genes and alleles. In this video, we will take a look at what genes and alleles are, as well as their functions. Oftentimes, when we want to know what something is, we want a very specific answer, like, what is an atom? An atom is literally the smallest unit of chemical matter. Well, that's really specific. Why can't all answers be that simple and that specific? Once these small units of matter begin to build on one another, we start to get into complex systems that work together to build other things. Well, genes are a result of that. Genes are made up of other things. So in order to answer the question, what are genes, we have to know what those other things are. In this case, we must first have a basic understanding of what DNA is. First, let me give a general answer to the question, what is a gene? Then we will go into a little more of the specifics. A gene is literally just referencing a very specific portion of your DNA. However, that specific portion is a full instruction to make something in your body. There are around 25,000 genes in the human body. So you can think of that as having 25,000 different complete instructions on how to make something. It may be helpful to think of it in this way. Let's say you have a giant instruction manual on how to build a car. Well, the manual is made up of very specific instructions, like what materials are needed, how to make the bolts, and all the way to what color to paint the car. Let's say that in this manual you read, use the metal titanium to make the bolts. You can think of that specific instruction as kind of like a gene. Here's what it looks like inside the body. Each cell in our body typically contains 23 pairs of chromosomes. We get 23 from mom and 23 from dad for a total of 46. Within these chromosomes are long strands of DNA. Well, remember, DNA is made up of 4 different nucleotides all paired together. The way in which they are sequenced is very important. Their specific sequencing tell our bodies what specific types of protein to make. Let's think back on our example of the giant instruction manual telling us how to build a car. Well, each set of instructions within that manual instruct us to do something very important and specific. The way that each set of instructions are written dictates how the car is built, assuming we are directly following the instructions. The words that the manual creator uses guide us and tell us exactly how to perform the work that needs to be done. Well, the same is true for our bodies. Our bodies are building proteins based on exactly how it reads the sequencing of nucleotides. In an instruction manual on how to build a car, you will see sentences that express a complete thought. And when you zoom further out, you will see paragraphs that express a complete instruction for a specific car part. Well, similarly, the entirety of our body's DNA represent our body's entire instruction manual. It's within these long strands of DNA where we can find our specific paragraphs or specific segments of DNA that tell us exactly how to build a specific part, or in our case, a protein, that make up the whole body. Well, that specific segment that tells us what specific protein to make is called a gene. So, now that you have a fundamental understanding of what a gene is and what it does, let's look at alleles. You may hear genes and alleles get talked about synonymously, or at the very least, when one is brought up, then the other is too. So what's going on here? What is an allele? An allele is a gene. However, here is how they are distinguished. Remember how we talked about how almost all of the cells of our bodies contain 23 pairs of chromosomes? 23 from mom and 23 from dad. Well, mom and dad are not the same. They do not have the exact same DNA sequencing. They certainly have a lot of the same DNA sequencing, but it is not all the same. All of the genes that their offspring will inherit, or segments of DNA that are identical, are called genes. The genes that you inherit that could potentially be different are referred to as alleles. An allele is a kind of gene that codes for observable traits, also known as phenotypes. A phenotype could be eye color, blood type, and so on. For example, you could inherit a gene or allele from dad that codes for the blood type A. Type A and type B alleles are always dominant alleles. So since you inherited blood type A from dad, you are guaranteed to have a blood type with type A in it. Let's say you also inherit type A from mom. You are going to have blood type A. So, does this mean that it is not an allele because both of the genes inherited from mom and dad are identical? No, because an allele just refers to genes that have the potential to be different, which are all phenotypes. So, it's always an allele if it is a gene that codes for a phenotype. We just looked at an example where a child inherited the same dominant blood type from each parent, but let's say that the child inherits a different blood type. Let's say that the dad gave the allele for type A and mom gave the allele for type B. Well, remember, type B is always dominant as well. So in this case, the child would have blood type AB. Blood type O is always a recessive allele. So in order for a child to have blood type O, it would need to inherit the allele for type O from not just one parent, but from each parent. Hey guys, welcome to this Mometrics video over DNA versus RNA. In this video, we will take an in-depth look at the structure and function of both DNA and RNA. To start, let's take a look at what DNA and RNA are, and then we will go back through and compare them. DNA stands for deoxyribonucleic acid and is a nucleic acid that contains the genetic instruction used in the development and functioning of all known living organisms. DNA is housed in the nucleus of the cell and works as a vehicle for storage and transferal of organisms' genetic information. DNA is a polymer that has a deoxyribose and a phosphate foundation with 4 nitrogenous bases: adenine and guanine, which are purine, and cytosine and thymine, which are pyrimidine. Now, adenine only pairs with thymine, and guanine only pairs with cytosine. DNA is a double helix structure and looks sort of like a spiraling staircase. The way that it spirals actually helps to give DNA added protection and stability. Let's take a look at exactly why. So, DNA has 2 strands, both located on the lateral ends, and these strands are twisted together like a spiraling staircase, which makes up the double helix. The lateral ends of these strands consist of sugar phosphate backbone of neighboring nucleotides that are bonded with one another. The phosphate molecule forms a covalent bond, which means it shares electrons with the deoxyribose sugar. These hydrogen bonds are what actually cause the DNA strand to spiral. The nitrogenous bases, adenine, guanine, cytosine, and thymine, are sandwiched between the 2 sugar phosphate strands. Each nitrogenous base is directly adjacent to another nitrogenous base, forming hydrogen bonds. Hydrogen bonds are weak bonds, but the bonds between the phosphate and sugar molecule are very, very strong bonds, which means they're very stable and can handle resistance. So basically, this phosphate sugar backbone's main job is to hold the nitrogenous bases that function as the coded set of instructions in place, and to protect them from outside elements. The spiraling of the sugar phosphate backbone adds 360-degree protection to the nitrogenous bases, rather than the nitrogenous bases being unspiraled and exposed. If it were not spiraled, then the weaker hydrogen bonds between the nitrogenous bases would be exposed to the surrounding elements, which could potentially break the bonds. RNA stands for ribonucleic acid and is a nucleic acid that executes the instructions given by the DNA. RNA is housed in the nucleus and the cytoplasm. The sugar in RNA is ribose. RNA is a single-strand chain of alternating phosphate and ribose units with the bases adenine, guanine, cytosine, and uracil bonded to the ribose. RNA molecules are involved in protein synthesis and sometimes in the transmission of genetic information. The primary role of RNA is to deliver the genetic information, or code, necessary for the building of proteins from the nucleus to the ribosome. This procedure stops the DNA from exiting the nucleus for the purpose of protecting the DNA. The production of protein is not possible without RNA. RNA is also crucial to the process of DNA transcription. An enzyme called RNA polymerase latches onto the DNA strand, then starts constructing a sequence of nucleotides to assemble a compatible RNA strand. There are 3 types of RNA: messenger RNA, or mRNA, ribosomal RNA, or rRNA, and transfer RNA, or tRNA. mRNA is transcribed from DNA, and it transfers the coded in structure for protein synthesis. rRNA is a part of the ribosome and controls the translation of messenger RNA into the proteins. tRNA carries the amino acids that follow the correct coding to the ribosomes to be attached into the proteins. Now, let's review and compare DNA and RNA. DNA has the sugar deoxyribose, while RNA has the sugar ribose. The difference between ribose and deoxyribose are fairly subtle. Ribose has 1 more OH group than deoxyribose has, and that is the only difference. Though the contrast is subtle, the difference changes the entire function. DNA is double-stranded. RNA, on the other hand, is single-stranded. DNA is more stable under extreme conditions, but RNA is not very stable due to it only being single-stranded. DNA and RNA perform different functions in humans. DNA is responsible for storing and transferring genetic information, while RNA directly codes for amino acids and acts as a messenger between DNA and ribosomes to make proteins. DNA and RNA base pairing is slightly different since DNA uses the bases adenine, thymine, cytosine, and guanine. RNA uses adenine, uracil, cytosine, and guanine. Uracil differs from thymine in that it lacks a methyl group on its ring. Hey guys, welcome to this Metrics video about the gastrointestinal system. The gastrointestinal system is also called the digestive system for an obvious reason. It's where all the facets of digestion take place. That means everything from the way food comes into our body, how our system breaks down that food, and how waste exits our body. It's a complicated but important process. So let's take a look at our gastrointestinal system in more detail. Our gastrointestinal system does more than just digest our food. Our bodies need nutrients from food and water for us to stay healthy and survive. Our gastrointestinal system breaks down food into very small pieces that can be absorbed by the body and used as a sort of fuel that we need to function. Major nutrients include proteins, which help fight infections and make healthy cells. Carbohydrates provide a source of energy. Minerals perform several functions, like keeping bones strong with calcium or providing an adequate supply of iron that helps transport oxygen in our blood. So yes, digestion is important, but our gastrointestinal system does so much more. So what happens once we ingest our food? Let's find out. We all have a gastrointestinal tract called the GI tract. This long passageway starts at the mouth and ends at the anus. Think of the GI tract as the entrance and exit point of our food. The GI tract is also called the digestive tract or the alimentary canal. It passes a number of solid and hollow organs, and we're going to take a closer look at those now. The GI tract has 3 solid organs: the liver, the gallbladder, and the pancreas. The mouth, esophagus, stomach, small intestine, large intestines, and anus are the hollow organs. Those hollow organs push food through our body via a process called peristalsis. It's the push and pull that allows food to move from one muscle to another. The muscle behind the food contracts and the muscle in front of the food relaxes, allowing for easy passage. That's the short of how food moves through our body. Now we'll take a journey through the gastrointestinal system and a more detailed look at its parts. We'll start at the mouth, the place that chews and ingests our food. After chewing, we swallow, and the tongue plays its important role by pushing the food into our throat. The epiglottis, a small flap, covers the windpipe so food doesn't get stuck there. And now that we've swallowed the food, the digestive journey starts. After we swallow, the food passes through the esophagus, and at that point, peristalsis begins. When the food reaches the end of the esophagus, it passes through the lower esophageal sphincter and then into the stomach. Have you ever gotten heartburn or acid reflux? You see, the sphincter is supposed to stay closed so food doesn't go back up into the esophagus. When the sphincter relaxes at the wrong time and food backs up, well, you get that heartburn feeling. But if everything goes as it should, the food has now made its way into our stomach, and that's where it goes for a nice digestive swim. Digestive juices coat the food in liquid and break them down so they can continue their journey through the body. But before we continue along our journey, we need to talk a little more about the stomach, a single organ that has 5 different parts. Those parts are the cardia, the part of the stomach closest to the esophagus, the fundus, which is located next to the cardia and is the upper part of the stomach, the main part of the stomach called the body, the antrum, the lower portion of the stomach, where food and liquid mix with digestive juices, and the pylorus, which lets food pass from the stomach into the small intestine. Remember the pylorus because that plays an important role in our next stop, the small intestine. Once in the small intestine, nutrients head into the bloodstream and waste starts the process of moving out. The small intestine has 3 sections that we'll briefly talk about here. The first section, the duodenum, is the smallest section. When food passes from the stomach, it enters the duodenum through the pylorus. The middle section of the small intestine, the jejunum, absorbs sugar and amino and fatty acids before it moves on to the final part of the small intestine. The last part, called the ileum, absorbs nutrients that were not digested by the jejunum. Those nutrients include carbohydrates, minerals, and fats. As food leaves the small intestine, it enters the large intestine. So if the small intestine is about 23 feet long, why is the large intestine labeled large if it's only about 6 feet long? You see, the large intestine at 3 inches has a larger diameter than the small intestine, which is just about an inch. The term large intestine refers to the larger diameter, not the total length. Large intestines takes food, breaks it down even further, and turns it into waste. There are several sections of the large intestine that participate in this process, and we'll go over those here. Large intestines has 4 parts called the ascending colon, the transverse colon, the descending colon, and the sigmoid colon. The ascending colon is the beginning of the large intestines and has 2 important parts: the cecum and the colic, or the ileocecal valve. The valve is at the end of the ascending colon and separates the cecum from the small intestine. The valve prevents food and other materials from flowing back into the small intestine. This process of fermentation takes place in the transverse colon. As food leaves the ascending colon and enters the transverse colon, fermentation further breaks down the food by removing water and nutrients. Once the fermentation process finishes, the remaining waste forms. Before we talk about the next step, here's another note about the transverse colon. 2 arteries, the medial colic artery and the inferior mesenteric artery, provide a constant supply of oxygenated blood to the transverse colon. Why? Because the blood flow keeps the intestine healthy and prevents illnesses like intestinal ischemia. Let's continue our journey. Food is moved from the small to the large intestine and has made its way to the transverse colon. The next stop, the descending colon. The descending colon stores food before it's emptied into the rectum. It's also the point at which feces start to become solid. From there, waste enters the sigmoid colon, and that's where waste solidifies. The muscular walls of the sigmoid colon contract to push feces into the next part of the gastrointestinal system, the rectum. The rectum acts as a holding tank for waste. The rectum holds feces until it's ready to be expelled. In other words, that's where waste stays until you're ready to use the bathroom. From there, the waste exits through the final stop in the gastrointestinal system, the anus. So that's the journey food takes. Now let's take a closer look at the enzyme production in the gastrointestinal system. Carbs, proteins, nucleic acids, and lipids are all major elements in biological molecules. Life can't exist if the body is missing any one of these molecules. So it's no surprise that they each play a role in the gastrointestinal system. Here's how. There are 3 enzymes that help break down carbohydrates. The salivary amylase enzyme in the salivary gland that helps break down starches and sugars. This process takes place in your mouth. The pancreatic amylase in the pancreas completes carb digestion and produces glucose. Lastly, the maltase enzyme in the small intestine breaks down disaccharides into single sugars. There are also 3 enzymes that help break down protein: pepsin, which is produced in the gastric gland, and trypsin, produced in the pancreas. Both help digest protein in food. Peptidases, produced in the small intestine, also breaks down proteins, but in this case, for the purpose of recycling amino acids. Nuclease and nucleosidase are 2 enzymes associated with nucleic acids. DNA and RNA are the 2 main nucleic acids. Both enzymes are produced in the pancreas. Lipids, those fatty and oily substances in the bloodstream, are broken down by lipase and bile salt. Lipase is found in the pancreas while the liver makes bile salts and the gallbladder stores it. Technically, bile isn't an enzyme. It's a salt that mixes or emulsifies lipid into fatty acids. We mentioned cells and patches, and they also play an important role. Chief cells located in the stomach convert pepsinogen to pepsin, which we already know helps digest protein. Goblet cells found in the respiratory and intestinal tracts secrete mucus. Parietal cells are crucial to the production of hydrochloric acid, which helps with digestion. Finally, Peyer's patches, the lymphatic tissues found in the ileum of the small intestine. protect the gastrointestinal tracts from pathogens. So that's our overview of the gastrointestinal system, the system that acts as an entrance for food and an exit for waste. As we noted, the gastrointestinal system also plays a role in breaking down nutrients that become fuel for our body. Hey guys, welcome to this Mometrics video over anion, cation, and the octet rule. Anions, cations, and the octet rule all have to do with atoms and their stability. Atoms will do anything within their power to be stable. Stability for an atom means that they have 8 electrons in their outermost shell. The electrons in the outermost shell are called valence electrons. Valence electrons are the things that the atoms want 8 of. And this is exactly what the octet rule refers to, the tendency of an atom to try and become stable by having 8 electrons in their outer shell. Atoms will go to great lengths to become stable. They will give electrons away, and they won't even think twice before they decide to steal electrons from another atom so that they will become stable. This is where ionic bonding comes in. Starting out, atoms are electrically indifferent or neutral. They have an equal amount of negative and positive particles. Negatively charged particles are called electrons, and positively charged particles are called protons. Well, when atoms lose their neutrality, they sort of go under an identity crisis. They are then called ions. An ion is an atom that has had an electron taken from it, or an atom that has taken an electron from another atom. So an ion is not neutral. It is either negatively charged because it has an extra electron, or it is positively charged because it is missing an electron and therefore has more protons than electrons. An atom that has had an electron stolen and in effect has more protons than electrons is called a cation. So a cation is a positively charged ion. An atom that has stolen an electron and therefore has a negative charge is called an anion. Let's look at some examples. Sodium, Na, is a very vulnerable element. It only has 1 valence electron. So for sodium, if it got rid of that electron, then it would be full in its now outer shell. But it would also be positively charged with 10 electrons and 11 protons. So in getting rid of an electron, sodium becomes unbalanced and positively charged, and is therefore a cation. Cations are noted with a plus sign superscript. This represents that it is positively charged. Chlorine, on the other hand, is 1 electron away from having 8 in its outer shell. So it wants very desperately to steal an electron. In stealing an electron, it becomes unbalanced and negatively charged. Therefore, it is an anion. Anions are noted with a negative sign superscript. This notation represents that it is negatively charged. Atoms can require or discard more than 1 electron at a time. Let's say we have the magnesium +2 cation and the phosphate -3 anion. Magnesium has an atomic number of 12. And remember, atomic number refers to the number of protons in the nucleus of an atom. So if it has an atomic number of 12 and magnesium +2 has a +2 charge, then that means it has 2 more protons than electrons. This means that it has lost 2 electrons. The protons aren't going anywhere. An atom will always keep the same number of protons that it starts with in its balanced state. The electrons are the ones changing. So if you see that an element is noted with a plus or minus, you can look at that number that follows, so in our case magnesium is followed by +2, and we can subtract 2 from our atomic number 12, and that will give us the number of electrons of that ion, which is 10 electrons. The phosphide ion has an atomic number of 15, so that is 15 protons, and it has a -3 charge. So it has gained 3 electrons. So we can add 3 to our number of protons, and that gives us our number of electrons in the ion. This means our phosphate anion has 18 electrons.
Hi, and welcome to this review of bonding and structure. In this video, we're going to take a look at how the bonding and structure of a substance relate to its physical properties. This particular subject is simultaneously simple and complex. The easy part is that, broadly speaking, we know that the physical properties of a substance are directly related to the bonding and structure on a microscopic level. So things like the melting point, boiling point, vapor pressure, and solubility of a substance can usually be understood by considering the molecular structure and bonding type. The complex part is that there are different types of bonds and lots of different chemical combinations, and there aren't hard and fast rules that dictate exact trends in physical properties. That's not too surprising given the huge diversity in materials. To get started, we'll review the different types of compounds and then consider a few physical properties and look at them from a molecular perspective. Keep in mind that there will likely be exceptions to all the rules I present today, but by the end, hopefully you'll have developed an intuition for how to assess a material based on its bonding and structure and have a good guess as to its physical properties. Let's begin with a quick review of the four primary categories of compounds. First, ionic compounds like sodium chloride are networks of cations and anions held together by strong electrostatic attractions. These are strong interactions and are generally pretty hard to break. Ionic compounds come in large arrays of repeating ions, and although written as NaCl, sodium chloride should be considered as a whole unit rather than pairs of ions. Second, molecular compounds like water, carbon dioxide, and caffeine are made of many discrete units of the same individual molecules, which are covalently bonded, but the units are held together by relatively weak intermolecular interactions like hydrogen bonds and dipole-dipole interactions. Third, covalent network compounds like graphite and diamond, both networks of covalent carbon, are held together by a continuous network of covalent bonds. This is in contrast to molecular compounds, where the bulk phases are held together by intermolecular interactions. Consequently, covalent network compounds have very different physical properties from molecular compounds, even though both have covalent bonds. Lastly, metals like copper or gold are held together by metallic bonds, or what is colloquially referred to as a sea of electrons. The valence electrons of each metal atom are more or less free to swarm around the lattice of metal cations. This unique type of bonding explains a lot about the physical properties of metals, which we will explain as we go. First, let's consider the melting points for these compounds. The melting point at standard atmospheric pressure will tell us what phase to expect when we encounter these compounds in our daily lives. As your experience might tell you, ionic compounds, covalent network compounds, and metals typically have melting points well above 25 degrees Celsius. In other words, all these are usually solids at room temperature. For example, sodium chloride has a melting point of 801 degrees Celsius, and quartz melts around 1670 degrees Celsius. This is a result of the very strong bonds holding the network of atoms together. You need lots of energy to disrupt ionic and covalent bonds, so the melting points are quite high. Metals are also solids at room temperature, with one notable exception being mercury, which melts at negative 39 degrees Celsius. Generally, the stronger the bonds holding the atoms together, the higher the melting point. Similar trends in boiling points exist for these compounds, but with much higher temperatures required for vaporization. Finally, let's consider the melting points of molecular compounds, things like water, carbon dioxide, and caffeine. As you can tell from our examples, molecular compounds can have very different physical properties. However, they generally have melting points well below those of ionic, covalent network, and metallic compounds because the molecular units are held together by weak intermolecular interactions. Typically, you can determine relative melting points of molecular compounds by assessing the weight and intermolecular interactions. Light molecules with weak intermolecular interactions, like carbon dioxide, will have relatively low melting points and exist as gases at room temperature. Conversely, heavy molecules with significant intermolecular interactions, like caffeine, will have higher melting points and are solid at atmospheric pressures. And then there's everything in between. Water is light, but has a really strong network of hydrogen bonds, so exists as a liquid at room temperature. Basically, to understand molecular compounds, you should always start by assessing their ability to form intermolecular interactions. Now that we've considered how bonding and structure impact melting and boiling points, let's consider another important chemical characteristic, solubility. As an upfront note, we will talk a lot about water in this section. After all, it is the universal solvent, but the word solubility doesn't just mean soluble in water. Solubility is the general measure of a solute's ability to dissolve in a solvent to form a homogeneous solution. And while the solvent is often water, it's important to keep in mind that that's not always the case. All right, let's dive in. When we think about solubility, the phrase like dissolves like often comes to mind. This is most relevant when discussing the solubility of molecular compounds. The like in the phrase usually refers to polar or nonpolar molecules, two categories that molecular compounds are broadly split into. Polar compounds have bonds where the electron density is unequally distributed among the atoms. They typically have a lot of heteroatoms, oxygen, nitrogen, halides, etc., which create areas of positive and negative charge. Strong intermolecular interactions occur when those areas in one molecule align with the opposite charges in another molecule. Thus, polar molecules tend to dissolve in polar solvents because of these attractive intermolecular forces. The process is energetically favorable. Conversely, nonpolar molecules are more or less neutral throughout and are mostly carbon and hydrogen. This means that they can only form weaker intermolecular interactions with other nonpolar and polar molecules. Let's consider an example. When you add oil to water, the water molecules, which are polar and form strong hydrogen bonds, are strongly attracted to each other. While the oil molecules are also attracted to the water molecules, their interactions with water molecules are so weak that they can't disrupt the network of water molecules and wind up sitting on top of the water. On the other hand, when you add ethanol to water, the two mix because ethanol is also polar and capable of forming hydrogen bonds and thus can compete with the attractive interactions between the individual water molecules. What's particularly instructive about this example of water, oil, and ethanol is that you need to consider solubility from both sides of the equation, the solute and the solvent. The system prioritizes the most attractive interactions, so it's not that the oil molecules don't like water. In fact, oil molecules have stronger intermolecular interactions with water molecules than with themselves, which is why a droplet of oil lays flat on a polar surface. It's that water likes water more than oil. Similar rules apply to solid molecular compounds. For example, vitamin C is water-soluble with all those highly polar OH bonds, whereas vitamin D is fat-soluble with a high concentration of CH bonds. This results in the two vitamins being absorbed and released from the body in totally different ways. Now let's consider the solubility of ionic, metal, and covalent network compounds. Since for the most part, all of these are solids at room temperature, we'll just think about their solubility as solids throughout this discussion. As you're probably familiar with, some ionic compounds are quite soluble in water. Table salt, for example, dissolves readily in water. However, other salts, like silver chloride, are essentially insoluble in water. Why is that? First, when salts dissolve, they split into their separate cations and anions, and they do so because of the many ion dipole interactions between the ions and water molecules. The water molecules form shells around the ions, stabilizing the raw charge. Importantly, salts don't dissolve because ionic bonds are weak. They are, in fact, very strong, as we've discussed. When salts dissolve, it's typically because the interactions with the solvent are, in aggregate, stronger than the ionic bonds. Another important consideration when thinking about solubility is the change in entropy that accompanies the formation of a solution. Because solutions are generally more disordered than the pure solute and solvent, dissolving a salt usually increases the entropy of the system. Thus, quantitatively, whether or not a salt dissolves is a fine balance between the energy associated with solvating the ions, the strength of the ionic bonds, and the change in entropy. These finer details we'll leave for now, but we do want to finish up this discussion with a quick question. As we know, sodium chloride dissolves readily in water, but would it dissolve in a nonpolar solvent like oil? Pause the video and think for a minute. Hopefully you answered no. The only reason sodium chloride dissolves in water is because the polar water molecules stabilize the system with many strong intermolecular interactions. Oil molecules, even those with a polar end, aren't capable of forming enough strong intermolecular interactions to stabilize the sodium and chloride ions. This is actually something you could test at home. Try dissolving salt in olive oil and water. You should see a pretty significant difference. Covalent network compounds are generally insoluble regardless of the solvent, as it's very hard to break the strong covalent bonds, and any resulting ions are too unstable for the solvation energy to make dissolution energetically favorable. Metals have a wide range of solubility. For example, sodium basically explodes when it's added to water. Lead only dissolves in acidic solutions, and platinum is nearly insoluble in everything save hot aqua regia, a mixture of hydrochloric and nitric acids. The reactivity of metals is connected to their electron configurations. Now, since we've been discussing the bonding and structure of these 4 different classes of compounds for quite a while, I want you to pause the video and give your best guess to this question. Which class of compounds do you think is best at conducting electricity? Keep in mind that the basic definition of electricity is the flow of electric charge. So take a second and consider your own personal experiences as well as what you've learned about the bonding and structure of the different compounds. Hopefully you came to the conclusion that metals would be good conductors of electricity. After all, electric wires are made out of metal. We can understand this from our discussion of metal bonding, which we described as a sea of electrons. The valence electrons of each metal atom are free to move around, which makes metals great at conducting electricity and heat. Conversely, covalent network, ionic, and molecular compounds are all generally poor conductors of electricity because the electrons are bound in covalent and ionic bonds and unable to move freely. But if you thought about answering that ionic compounds would be good electrical conductors, you weren't too far off the mark. When ionic compounds are melted or dissolved in solution, they are great electrical conductors because there can be free movement of charge. And lastly, let's consider how bonding and structure are related to some of the other physical properties of a compound, like how hard or malleable it is. Ionic compounds are hard and brittle. They can't be molded because if you were to shift the ions, you may align like charges, in which case the ions would strongly repel each other and break along a clean line. On the other hand, metals tend to be malleable. The sea of electrons holding them together can shift easily and prevent repulsion between neighboring cations. Consequently, we're able to work metals into complex shapes. Covalent network compounds are typically hard and unworkable, while molecular compounds run the gamut with their physical properties dictated by the specific intermolecular interactions. Well, we've covered a lot today, but have only scratched the surface of how materials connect to their physical properties. Hopefully, you've seen that the chemical and physical properties of a compound are always connected to what's happening at the molecular level. Thus, to best understand material characteristics, we need to know how the atoms are bonded and about the distribution of charge.
Hi, and welcome to this video on the states of matter. A state of matter is a distinct form in which matter can exist. Today we'll be looking at the 4 states that are observable in everyday life: solid, liquid, gas, and plasma. Let's start with solids. The particles in solids are tightly packed, usually in a specific geometry that repeats itself, called a crystalline structure. There are also amorphous solids where the particles are not in a replicated geometry. There are 4 types of crystalline solids, all of which are held together by different forces. Ionic solids, like table salt, which is sodium chloride, are held together by electrostatic attractions. In covalent network solids, like graphite or diamonds, the atoms are connected through covalent bonds. Molecular solids, like ice, are held together by many intermolecular interactions. Metal solids like copper are held together by metallic bonds, or what is colloquially known as a sea of electrons. As you know from the examples, solids can have very different physical properties, all of which relate back to how the individual particles are connected. However, as solids, they share some physical properties. Solids are usually the densest of the 4 phases. Water is a notable exception, and are therefore nearly impossible to compress. The particles are locked into position and have no translational kinetic energy. However, the atoms or molecules can still be vibrating and rotating in place. Solids hold their shape, rather than conforming to their vessels. Liquids are also a condensed phase. However, in contrast to solids, the particles have some translational kinetic energy, which means they have freedom to move. The particles still hold on to each other, but have enough energy to continually break away from each other and form new interactions with adjacent particles. This makes liquids flexible, allowing them to flow and take on the shape of a vessel. While liquids are usually less dense than solids, they are still packed tightly enough that they cannot be compressed. In other words, they have a definite volume. Substances transition to the gas phase when the particles have enough energy to completely overcome the interactions holding them together. Consequently, gas particles have lots of translational kinetic energy and are constantly zooming around, vibrating and rotating. They expand to fill whatever container they're in, and the many tiny bombardments from the particles exert pressure on the container wall. Because there's so much space between particles, in contrast to solids and liquids, gases can be easily compressed. Finally, plasma is a highly energetic gaseous mixture of charged particles, though overall the plasma is neutral. It is considered separate from the gas phase because it has unique physical properties. Like gas, plasma expands to fill its container and is compressible, but very different from standard gases. Plasma has high electrical conductivity. This is because some of the charged particles moving around freely are electrons. While naturally occurring plasma is rare on Earth, our stars are made of plasma, making it the most common phase in the universe. Now that we've covered each phase of matter separately, let's consider them together. You'll notice that as we progressed from solid to liquid to gas to plasma, the internal energy of the system increased. Consequently, it's often possible to prompt a phase change by simply increasing the temperature of the system. By providing thermal energy, we can break the forces holding particles together and increase the translational kinetic energy of the particles, which typically produces a phase change. The amount of heat necessary to undergo a phase change depends on the type of material. Returning to the 4 solids we discussed, usually the forces holding ionic, covalent network, and metal solids together are much stronger than the forces in molecular solids and consequently have much higher melting points. For example, sodium chloride and copper melt at 1,474 degrees Fahrenheit and 1,984 degrees Fahrenheit, respectively, versus ice, which melts at 32 degrees Fahrenheit. In addition to temperature changes, you can prompt a phase transition by adjusting the pressure. The way a substance changes with pressure and temperature is often mapped out in a phase diagram. While these are material-specific, in general, substances tend to be solids at low temperatures and high pressures, gaseous at high temperatures and low pressures, and liquid at intermediate temperatures and pressures. Since most of us encounter only standard pressure and temperatures, we often think of materials as always being in a specific state of matter. For example, most of us think of salts and metals as being solids. But as you can tell from the graph, it is possible to convert between phases. It just might require extreme temperatures and pressures. Let's finish with a quick review. We defined the 4 states of matter and connected their atomic structure to their bulk physical properties. We noted that the internal energy increases when transitioning from solid to liquid to gas to plasma, and discussed how the phase of a material changes with temperature and pressure.
Hey everyone, welcome to this Mometrics video over the 4 laws of thermodynamics. Thermodynamics is a branch of physical science, or physics, that focuses on the correlation of heat and all other forms of energy, and consequently, all forms of energy that relate to one another. Well, the 4 laws of thermodynamics work to define the foundational physical quantities that mark thermodynamic processes when at thermal equilibrium. These foundational physical quantities include temperature, energy, and entropy. Now, let's take a look at how these laws are defined. The 0th law actually came after the first 3 laws of thermodynamics, but it was actually so fundamental and provided a basis for the other laws that it got the name 0th. That way, people knew the order in which they should learn or think through the other laws. So, what does the 0th law actually state? Let's take a look. Let's say that you have 2 systems, A and B, that are connected to each other with some sort of barrier between them. Well, the heat or energy of A will transfer over to B, or vice versa. The energy of B will transfer over to A. This is assuming that the wall is diathermic, meaning that heat can be exchanged, as opposed to an adiabatic wall, which would not allow for heat to pass. Once heat or energy is transferred between 2 systems such that there is no more heat being transferred or the transference of heat stops, then the systems have reached equilibrium, more specifically thermal equilibrium, since we are talking about heat. Now, let's say that we have another system, A and C, and they also reach equilibrium. Well, since A is in equilibrium with B and A is in equilibrium with C, then when you take B and C and place them together with a diathermic wall, then there will be no transference of heat. This is because the 2 systems are already at equilibrium together. To say this mathematically, you could say if A equals B and A equals C, then B is equal to C. That's the 0th law of thermodynamics. For some of you, this may seem like common sense. To others, it may seem counterintuitive, especially if you are super familiar with Boyle's law. Regardless, this is what the 0th law shows us, but it gives us much more than that. The illustration you just saw tells us something. What is that? What is that physical quantity which determines that the 2 systems will reach equilibrium and the transference of heat will stop from one system to the other? Well, it's this very physical quantity that was discovered later. There were already 2 laws of thermodynamics in place, the 1st law and the 2nd law. Well, in these 2 laws, the term temperature was used, which was thought of as this degree of hotness, basically. While the term temperature was being used in the 1st and 2nd law, no one could actually define it. There wasn't really a way to define temperature within thermodynamics. So, the 0th law was made so that temperature could be defined. So, how is temperature defined? Here we go. If there are 2 systems in contact with each other, then the direction of heat flow tells us that one body is at a higher temperature and the receiving body is at a lower temperature. So, temperature is the physical quantity which determines the flow or direction of heat. So, temperature decides if heat will flow from A to B or from B to A. Let's review that real quick. First, 0th law says that if 2 systems, C and B, are in thermal equilibrium separately with a system A, then C and B are also in thermal equilibrium with each other. Secondly, the physical quantity defined in the 0th law is temperature. And lastly, Temperature can be defined as the physical quantity which determines the direction of flow of heat and is the one that is equal in a state of equilibrium of two systems. That was the 0th law of thermodynamics. Now let's take a look at the first law of thermodynamics. Here's what the first law of thermodynamics states. Heat energy given to a system is converted to its internal energy and work done on that system. To put this even simpler, the first law of thermodynamics just states that energy cannot be created or destroyed. It can only be transformed. Let's look at that mathematically. Internal energy represented by delta U is equal to heat energy plus work done. This equation is known as the first law of thermodynamics. But I'm guessing this still doesn't make sense to you. I don't blame you. Let's look at it this way. You have a system. Everything on the outside of that system is the surroundings. Well, energy can flow either into the system or out of the system. There are two ways in which energy can flow in and out of a system, which is through heat and work. So let's say that heat flows into the system. When heat flows into the system, then the system accumulates or gains energy. That energy is called the internal energy of the system, which is where we get our symbol U. The surroundings can do work on the system. So heat and work are the two ways in which a system can increase its internal energy. If the system's surroundings were to do 150 joules of work on the system, that means that the system's internal energy increases by 150 joules. And since the change is positive, delta U goes up. And since we know that energy cannot be created or destroyed, only transferred, the energy had to come from somewhere, the surroundings. So if the system's internal energy increases, then the energy of the surroundings must decrease. Well, energy follows this same principle. It cannot be created or destroyed. It's simply transformed or transferred from one place to another. The next law we will look at is the second law of thermodynamics. Now, the second law of thermodynamics might seem like an odd law. Why? Well, firstly, because the second law of thermodynamics doesn't tell us what can happen or what is possible. It only tells us what cannot be done or what is not possible. Another reason it might seem odd is because there are several ways to state it. The second law of thermodynamics states that the efficiency of a process can never be 1 or 100% of any machine. If a machine or process operated with 100% efficiency, then all of the heat or energy put into a system would be completely converted into work done, and there would be no heat rejected out. The second thing the second law of thermodynamics states is that unless you apply a pump into a system, there is no way to send heat from a cooler source to a source at a higher temperature. Another popular way to state this law, as put by Clausius, is no process is possible whose sole result is the transfer of heat from a colder object to a hotter object. Put very simply, the second law of thermodynamics tells us that heat will never flow from a colder object to a hotter object unless you use a pump to do so. Like how a pump is used to cool the inside of a refrigerator. In every other case, however, heat always goes from the object at a higher temperature to the object with the lower temperature. And the other thing it states is that within a system or machine, heat or energy will never be 100% converted into work done. So if I put 100 joules into a system and I get more than 100 joules out of the system, then one of three things has happened. 1, I lied. 2, I miscalculated either my input or my output. Or 3, I just turned all of physics and science on its head and defied all of it. The third law and last law of thermodynamics defines absolute zero and brings together the concepts of entropy and temperature from the latter laws. The third law states that the entropy of a perfect crystal approaches zero at a temperature of absolute zero. We can't actually achieve absolute zero experimentally, or at least you probably won't. This is because we know from the second law that heat is spontaneously moving from a warmer body to a cooler body. So the object that you are trying to get to absolute zero will be constantly taking in heat from its surroundings. I said that the lowest possible entropy will only occur in a perfect crystal. Well, a perfect crystal is a structure where all of the atoms are identical and are positioned in perfectly symmetrical ways. If there are any imperfections in the crystal, then that imperfection carries an energy with it, which does not allow for the minimization of entropy. Another important note is that if we can't get to perfect zero, then there will never be complete stillness in our universe, because we will always have some sort of motion due to thermal energy.